to obtain the necessary parasites for a viral screening experiment. Clarita is going to dissect this Pihocoris or firebug, obtain his gut, and seed it into a culture media. So first he will catch it and put into 70% ethanol, where he will drown to death. So usually we wait until it stops moving in the bottom of the beaker. This tells us that the animal is already dead. We should always be careful with this and watch it closely, because if we leave it too long, the animal can swallow ethanol and this will kill the parasites inside its gut. After drying the alcohol, she will lay the bug into the slide and start the dissection of the abdomen of the insect. She will now separate the head from the abdomen. Or try. And now she'll do a sagittal cut in the abdomen. Now, she will add a small quantity of physiological solution to the extracted gut. This will help increase the volume and facilitate the visualization of any possible parasites on the microscope. Now she'll check carefully for the presence of flagellated trypanosomatids swimming in the liquid. So now she will seed the parasites into a small volume of culture media, where they are going to stay for at least 24 hours before being finally transferred to bigger volumes. After staying around 24 hours in this tube, they are going to be transferred to a bigger volume of culture media, where they are going to grow for around 4 or 5 days until they get to the correct density of 1/10 to 7 cells per ml. These cells are going to be harvested in a centrifuge, spinning down at 1000 xg for 10 minutes, and then we start the RNA extraction using the RNA-Zol kit. So we use the direct-to-Zol kit to extract the RNA. This kit is based on a column system, where the RNA mixed with ethanol binds to this column, and then the RNA can be eluted in water or any buffer of your choice in the end. So in here I'm using a column-based kit to purify RNA from the cell cultures that you just saw being obtained. This kit will elute the RNA into any buffer of your choosing in this last step. In my case, I choose water because I will do 2 subsequent enzymatic treatments in order to isolate the double-stranded RNA from the virus that we are looking for. So, after adding the desired amount of water, I'm going to spin this for 1 minute at 10,000 xg. So now to 10,000 x. And this RNA is going to be measured for its concentration in the nanodrop. I'll show now. So basically this is a nanodrop that is going to use two wavelengths of UV light, 260 and 280, to measure the concentration of RNA in the sample. This is just water for a blank. Now I'm going to add the RNA here. 1 microliter of RNA, which is 1/1000 of 1 ml. Who is going to be watching? And now I can finally measure what will appear here, the concentration. Okay, so we got RNA in decent concentration and in excellent quality, as you can see by the ratio. Our RNA will be treated with two different enzymes. The first one is DNase 1. It has its buffer and enzyme. DNase 1 will remove any contaminant related to DNA from our sample. In all extractions of RNA, you tend to have a DNA contamination, even if you use the best column. This is why we do it. And after, we treat it with S1 nuclease, which is an enzyme that can destroy any nucleotide that has single-stranded form. So it will digest any messenger RNA, tRNA, or single-stranded DNA too that is present in the sample. And with this, we are going to end up with just the double-stranded RNA that is present in the replicative phase of the virus. So we use 50 micrograms of RNA for each treatment. So in this case, we will use 20 microliters of my RNA. Okay, so you usually take the RNA and any necessary water. In this case, I will use exactly 20 microliters. So 20 microliters. Then I will add the necessary amount for 25 microliters of total reaction of buffer. So 2.5 microliters in the end. Okay, 2.5. And I'm going to use 1 microliter of DNase 1. This one is 2000 units per microliter. So by our calculations, it should be enough to treat 550 micrograms in 1 hour of treatment. This sample will be held at 37 degrees in a dry bath for 1 hour. After this 1 hour, I'm going to take it, spin it down, and add 1 microliter of S1 too. This takes less time, approximately half an hour to 45 minutes. And after I finish this treatment, I can finally run this sample into electrophoresis gel and agarose and see if I indeed have double-stranded RNA there or not. So after enzymatic treatment, RNA can be run into electrophoresis, into an electrophoresis agarose gel. And the double-stranded RNA bands can be visualized if resistant.
So here we have a machine for qPCR, actually this one. And here you also can see the computer which is connected with this qPCR machine. This qPCR machine we use to measure any DNA which level we want to see. For example, we can detect level of virus in our cultures or level of any other DNA. For that we use this kind of plates. Here we usually load our sample, and then when we prepare our plates we can load this plate here. And as I said, in every well we have our sample with some culture. Then this machine just eats this plate, and then in every well we can see level of any DNA which we want to detect. After this qPCR we can see result which is visible here. This all our samples which we detected. For example, this 3 replicates of our sample, and it shows that for example, level of our DNA started to increase after cycle 20 or after, for example, 10 minutes. Then we can download our data from this machine, and after some analysis we can get result which will look like this. I hope it's visible. So, for example, here we had some amount of samples, and in biggest part of our sample we could detect that we have virus in our cultures, but in 4 other cultures we could see that we do not have virus in them. So that's in principle how it works. And basic of this that we have special chemicals which we call like special primers, and these primers can recognize any sequence which we want. For example, a sequence of our virus which we want to detect. Then these primers usually bind to this DNA, and then with special chemicals it produces fluorescence, and this machine can detect this fluorescence, and as a result we can see how many viral particles we have in comparison with some other gene.
When we get raw data from the sequencer, we process it using command line tools in Linux environment. To process big data, we need to use cluster computers, because normal computers are not powerful enough. We can use various tools to look for genetic features of interest. In the end of the data processing, we can get the sequence of the virus we study, and using electron microscope we can even make a picture of the virus.